Hello, dear students. In this lecture, we will discuss how to prepare a tissue culture medium. So, a tissue culture medium is a nutrient medium which is used for the growth of plant cells and tissues uh, in vitro conditions. A basic tissue culture medium consists of macronutrients, micronutrients, vitamins, a source of iron, source of carbon, that is sucrose, a gelling agent, which is generally agar-agar, and growth hormones like gibberellic acid, auxins, cytokinins, etc. So, what are the constituents of our tissue culture media? First constituents are macronutrients, which are the primary nutrients required for the growth of plants, and they are required in large amounts. Examples are nitrogen, phosphorus, potassium, calcium, magnesium, etc. Then the second component is micronutrient. The micronutrients are required in small amount, uh, and these are also uh, known as the elements which are required in trace amounts, which includes iron, manganese, zinc, copper, boron, molybdenum. So here in this slide, you can see that every line is referring to MS medium, because MS medium is the most widely used medium in tissue culture. You can make any media like LS medium, Gambox medium, NP medium, Nish medium, but we will be referring to MS medium everywhere because that is the most widely used medium in tissue culture labs. Now we were discussing the third component. So third component is vitamin. So the source of vitamins are added in any medium, and these vitamins include vitamin B1, pyridoxine, that is vitamin B6, nicotinic acid, riboflavin, etc. The fourth component is an iron source, which is generally uh, given in the form of EDTA, iron EDTA, and in this iron remains available up to a pH of 8. So besides the first four components, that is macronutrient, micronutrient, vitamins, and iron source, nutrient media consists of sucrose in the percentage of 3%. So 3% sucrose is used as a source of carbon for the growth of plants, and also to if we want to make a semi-solid medium, then we need a gelling agent or a solidifying agent. So agar-agar is the most widely gelling agent used in plant tissue culture, and the percentage which is used for solidifying or making the medium semi-solid is 0.8% agar-agar. If we do not want to make a semi-solid medium and we just want a liquid medium, we do not add agar-agar. Then, along with this, in a plant tissue culture medium, growth regulators are added, which are actually hormones, and these hormones are generally added for the development of roots and shoots, and these are cytokinins and auxins, gibberellins, etc. You can see here, this is a tissue culture media, and this tissue culture media is poured in Petri plates, and these tissue culture media is poured in test tubes, and these test tubes, you can see that the medium is poured in a slanting position. So this medium is present in a slanting position to increase the surface area of tissue culture medium, so that our explants gets what is the procedure of preparation of a tissue culture medium? So there are two ways through which we can make tissue culture media. First is using a stock solution, where the concentrated stock solutions are used, and we prepare stock solutions, and we store them in refrigerator. Whenever the requirement is there, we use them. And the second method is that we prepare fresh media every time, using the commercial salts available. And so first we will discuss how to prepare tissue culture media using stock solution. So as we have just discussed that, for when we prepare stock, so we can stock solution we can keep for a long time in refrigerators. So generally, when the macronutrient stock solution is formed, it is made in the concentration of 10x. And when we make micronutrient stock solution, it is made in the 100x concentration. So using this macronutrient stock solution and micronutrient stock solution, we can take the required amount of macronutrients and micronutrients according to the volume of tissue culture media which we are using. Then generally the iron source, that is iron EDTA, is added fresh, and vitamin stocks we can keep prepare, and we can keep them, and we can add them in the media whenever we are making media. So in this slide, you can see table 2 and table 3. In table 2, you can see that for the first column is the constituent, the second column is the concentration which is given in milligrams per liter of MS medium, that is your Murashig and Skoog medium. Fourth is your stock solution, and third column is your stock solution, and fourth is your volume of stock. So what does that mean? It means that suppose we are taking micronutrients, and suppose we have made micronutrients in 10x stock solution. So now the macronutrient composition, that is of stock solution 1, it consists of ammonium nitrate, potassium nitrate, magnesium sulfate, potassium hydrogen sulfate. And if we suppose we are making 1 liter of MS media, then we require 1650 grams of ammonium nitrate, 900 grams of potassium nitrate, 370 grams of magnesium sulfate, and 170 grams of potassium hydrogen sulfate. But suppose we have made a stock solution, so we don't have to take it individually. We will just make a stock solution first, using these using taking these quantities in with respect to 10x stock. So for making a stock solution, we will take 16,500 grams of ammonium nitrate, 19,000 grams of potassium nitrate, 3,700 grams of magnesium sulfate, 1,700 grams of potassium hydrogen sulfate, and we will make up to 1 liter. So from this 1 liter stock solution, we will take 100 ml whenever our requirement is there. Suppose we want to make 1 liter of MS medium, so from this stock solution we will take 100 ml, and that 100 ml will consist of these required amounts of stocks of our macro nutrients. Similarly, the macronutrient stock solution 2, we will take 100 ml. Suppose we have make stock solution of 10x, so we will take 100 ml from this, and that will be our that will fulfill our requirement of 450 grams of macronutrient stock solution in the tissue culture media. So then you have micronutrient stock solution 3. So as we have discussed that we micronutrient stock solution we make in the concentration of 10x, and from that we 10x solution will take 10 ml to fulfill the requirements for 1 liter milligrams 1 liter solution of MS media. Then iron EDTA, we have discussed that it is added fresh. Then vitamins, these are the vitamins, so we can just take the stock solution of vitamins and we can add directly to the medium. And then we have discussed that sucrose we require 3% concentration. So suppose we are making 1 liters of MS solution, so we require 30 grams of sucrose, and agar is used in the concentration of 0.8%. So if we are making 1 liters of MS solution, we will take 8 grams of agar. So for and a pH is adjusted using 0.1 normal NaOH and 0.1 normal HCl to 5.8. And then we can also add the growth regulators. So growth regulators, we have discussed that they are cytokinins, auxins, gibberellins. So we have to see whether these are we can dissolve in water or some other solvent, and accordingly we can use the or add the growth regulators in our basal media which we have prepared using macronutrients, iron, ADTS, sucrose, and agar-agar. So as you can see here, that what are the cytokinins? BAP is your 6-benzyl adenine, 2IP kinetine, zeatin, and TDZ. So we know that the first four cytokinins we can dissolve in water, and TDZ is dissolved in ethanol, and accordingly, according to our requirement, we can take we can add the cytokinins or auxins in our basal medium which we have just prepared. Some Most of these growth regulators are autoclavable, and sometimes we have to use micropore filter, and we have to add after making the media in after autoclaving the media in tissue culture lab in laminar flow. So what are the steps involved in the preparation of the medium? So as we have just discussed, first we prepare a stock solution using tables 2 and 3. So these tables are given in any tissue culture book, and in a sequence we add micronutrients, macronutrients, iron, EDTA, vitamins, and then we make the final volume of the medium with distilled water. Our final volume can be 1 liter or 500 ml according to our requirement. Then we add the next point is that we add sucrose in the concentration of 3%, and after adding sucrose we mix well and adjust the pH using pH meter in the range of 5.5 to 5.8, and 5.8 is the perfect pH for any tissue culture medium. At the end we add 0.8% agar, heat it, and then we autoclave it in the autoclave. So there are two ways. First, we can just melt the agar and pour in the test tubes, or what we can do that we can and later we can autoclave the tissue culture test later we can autoclave the test tubes in autoclave, or what we can do that first we make the media and then we autoclave it, and then we pour in laminar flow. So the second method is quite cumbersome. So usually we make the media, we pour in the test tubes, and then we directly autoclave, and then we make the slants after cooling down. So once we autoclave the medium at 121 degrees Celsius for 15 to 20 pounds, then after cooling down we make the slant, and then our medium is ready. So this is a semi-solid medium which was prepared using agar agar. If we want to prepare liquid medium, we can omit the step of adding agar agar. Now the second way of making any medium is that we directly take the we buy the salts from the commercial supplier, and then we can make the medium directly using salts whenever we are making the medium. So here you can see the picture that this is a high media salt of Morashig and Skoog macro elements, and they are saying that if we take this macro elements which are actually macro elements or macronutrients, then we have to take 4.2 grams per liter. Similarly, they have micro elements or micronutrients. And so in from when we are using the commercial salts, we can just take according to the instructions which are given on the bottle, and we can prepare the media. Like for here you can see that if we are taking this salt, this is a macronutrient, so we take this salt, we add sucrose, we add agar agar according to our requirement, and then we adjust the pH, and then we autoclave it, and the medium is ready. So this is when we do not have stock solutions, and we directly go for the media which is already available in the form of commercial salts. So here in this picture you can see that these are tissue culture media which are poured in test tubes, and the explants have been cultured. And these, there are different explants you can see. Some are seeds, some are leaf explants, and you can see that the media is poured in test tubes and the media is slanted to increase the surface area so that more number of explants can be cultured on the media. So I hope students you have understood the topic, and you can refer any good tissue culture book to understand it better. So tissue culture book like Bhojwani and Razdan, Bhojwani and Dantu, any book you can refer. So thank you students, and let us meet in next lecture.